So, welcome everybody to our webinar on fish migration and hydropower plants, which should give you some insights on the effects of turbines on the fish that are migrating through them, and also evaluate some mitigation measures that are undertaken. So this webinar is mainly organized by the project Life for Fish, and mainly by Iman Ben Ammar, who is a postdoc at the University of Namur and who is working for the project, and also by the Institute of Life, Earth and Environment, which is one of the research institutes at the University of Namur. So just one organizational remark. We will have a short introduction to the project and then 2 talks that are about 20 minutes or 25 minutes long, with some time after the talks to discuss. And I will ask you to present your questions in the chat modus, and I will ask the question after the talks and they will be answered at that point in time. So now I leave the word to Robert Mondicchi, who is also a member of the University of Namur, to give us the introduction to the project Life for Fish. Thank you, Robert.
Welcome to everybody. I will introduce you briefly to the objectives and summarize the main actions of the project. The Life Fish project is coordinated by EDF Luminis, which is one of the largest producers of electricity in Belgium. And as partner, we have 2 universities, the University of Namur, the University of Liège, and Profish, which is technological in hydrological management. The main objectives of the project, I will share my So briefly, the main objectives of the project is to contribute to the preservation of aquatic biodiversity in the Low Meuse River by improving the ecological continuity while maintaining a cost-effective hydroelectricity production. As you understand, the coordination of the project is hydroelectric production, which is EDF Luminis, to join the contribution of fish conservation and the production of green energy. As specific objectives, and first, the project focus on Atlantic salmon and European eels. And the specific objective of the project are to characterize the stock and migrating fish populations, to describe the downstream routes and design a predictive and reliable downstream model for each species. All the objectives is to select sustainable and appropriate solutions, which will increase the escape flow, but also the welfare of fish through the hydropower facilities. As main actions to achieve these objectives is the characterization of physiological and health status of the stock population and migrating fish after passage through the turbines, and the application of hydraulic modeling to the hydraulic works in order to design innovative solutions, but also the definition of hydropower management strategy based on the designed downstream migration models using, for example, radium telemetry, and to switch the production of electricity during the migrating, the downstream migration of fish. And as concrete actions, the projects deal with installation of sustainable or fish-friendly turbines, and also for installation of repulsive behavioral barriers, but also safe downstream fish passage. And as a final goal of these all actions is to increase the survival after the passage to the turbines along the low Meuse River to up 90% for salmon smolt, up to 90 for silver eels. So now Iman will present the result that we already obtained on the actions concerning the physiological, human, and health characterization of the migrating populations.
Okay, thank you very much, Robert, for the introduction. And Iman, I give the word to Iman, who will tell you now about the impacts of turbine on salmon smolts and European eel.
Hello, everybody. Let me just very quickly share my screen. So, today I will share with you our results about the turbines' direct and delayed impacts on migrating Atlantic salmon smolts and adult European eels that were acquired during our experimentation in the framework of the Life for Fish project. It can be a little bit lengthy because we have results on smolts and on adult eels, but I hope that you will follow me and you can have all your questions put on the chat after the talk. So the Meuse River is a very it's a very heavily modified water body with dams, hydropower plants, where and many other things, and it is in the same time home to 10 highly migratory diadromous fish species and more than 30 non-diadromous fish species. Why it doesn't talk? Atlantic salmon is a species that migrate from the growth area in the ocean to upstream to the river in order to fray, and after the smolt will migrate back to the ocean for the growth area. And during its downstream migration, Atlantic salmon smolts can be faced with many hydropower plants that have an effect on them. And it is already known that the disruption of hydrological connectivity of the rivers led to severe reduction and even the extinction of some strain of Atlantic salmon in Europe and America. And in the Wallonian Meuse River, we don't have anymore any wild population stock. All the population that are in the Meuse River, in the Wallonian part of the Meuse River, come from restocking program. The European eel is a critically endangered species that migrate from growth area in the river to the Sargasso Sea to reproduce, and during it is and come back after upstream to growth. And during the downstream migration, the adult silver eels can face the hydropower plants. And we already know for the European eels that multiple factors, including habitat loss and migration barrier, that can be dams and also hydropower plants, led to a strong decline in the wall distribution area for the European eels, and this species is already considered as critically endangered from 2008. So what does the hydropower plant? They impede the free movement, they prevent and limit the access to functional habitat, they increase the fish stranding, and they also lead to mortality that are directly linked or indirectly linked to the passage through the turbine or over the spillways. The area of the Life for Fish project that you see here, the area of study, is the Wallonian part of the Meuse River, and it includes 6 hydropower plants. What happens when the fish faces a turbine? Generally, fish are facing strike from mobile and non-mobile part of the hydropower plant. They also face sudden speed and pressure change, shear stress and cavitation. What is known is generally the survival rates and the damage rates from many studies that were already conducted. Those studies can be telemetric studies that follow fish that are migrating downstream, or direct impact studies that are done by putting the fish, injecting them directly in the turbine and estimating after the impact in terms of survival and damage rate. What we know is that in Atlantic salmon smolt, the survival rates vary from less than 60% in some commonly used turbine to more than 90% in some environmentally friendly turbines. The eels survival rate vary from less than 30% to more than 95%, depending on manufacturer, and those factors include the turbine's type. And it is very important to know that the size of the fish is an aggravating factor of the damage and the mortality occurring during the passage through the turbine. But what we don't know at all is how the surviving and injured fish are affected by this passage, because we know for fish that are already dead, the fish that are already injured, we know that they exist, we can see them, but nobody already investigated what happened for those that didn't get injured. So because we wanted to know what happened for the fish that are not injured and that survived the passage through the turbine, we focused on studying how the passage through the turbine will affect the physiological condition. It includes the stress status, the metabolism and hormone regulation, and the health and human status of the fish, with prospecting the innate immune system and oxidative stress defense in both species, so Atlantic salmon smolt and European eel adults. And our assumption is that the stress that occur during the passage through the turbine can lead to a disruption of the human system, and with the injuries and the presence of pathogen, it can make the fish more vulnerable and lead to indirect mortality after the passage, and decrease the success rate to escape to the ocean. So we conducted 2 big experiments, one at Onden site and another one at Grand Malade site. Both sites are hydropower site owned by Luminous, which is the coordinator of the project, and both the experimentation were done on both species. For Atlantic salmon and European eel, the procedure was the same. It was the release of the fish upstream the turbine and the recovery of the fish with a big net after their passage through the turbine. It is a technology of direct impact that is already done and used by Profish. And that is why we collaborated with Profish to make those experiments. And what we do is putting the fish to acclimate before the experiment in tanks that are alimentated with the river water. After, in the day of the experiment, we will the fish will be caught, put in a wetted tube, and directly release it, as you can see in the C picture, directly release it in front of the turbine, or for the control group, they will directly release it in the net. The turbine is set as its maximum intake during the experiment. So we have two groups, one that go through the turbine and another one that go directly to the net. The first group is the HPP group, the second one is the control. And in the experiment of then we injected 2 times 180 fish smolt in the HPP and 180 fish in the control group. We recovered them, we recorded the survival rate, and we determined the type of injuries, and the dead fish and the very heavily injured fish were euthanized, and the ones that were injured were put back in their corresponding tanks. 24 hours after the injection, we sampled fish for blood and organs, and we did it again 72 and 120 hours post-injection. And we wanted to determine the plasma cortisol and glucose levels and stress-related gene expression, mainly HSP70, some carbohydrate lipid metabolism genes, and hormone regulation genes. In the same time, we determined on the plasma the peroxidase and SCG50 complement activity, and the and in the organs, which was the spleen, we determined the immune system related gene expression, mainly IgM, lysozyme G, complement, MPO, and COX-2. For the eels, so the procedure was exactly the same, but we had two, actually today I will present the results of two experiments, one in Grand Malade site and another one in Antenne site. And we injected so eels directly in front of the hydropower plant or directly in the net, and simply recovered them, recorded the survival rate, did the damage rate, external damage rate, and sampled them after a 24-hour, put them back in their tank and sampled them after a 24-hour post-injection and 144-hour post-injection. And in the same, at the end of the experiment, we did X-ray on the eels in order to determine the internal injuries. And what is the difference between the both sites? It is in Grand Malade, it is a straw-flow turbine that is a low head one. The head is 3.8 meters and a less veloce one. In Antenne site, it's a bulb turbine. So both of them are Kaplan variant turbine with a higher head and that higher velocity. And we sampled eels only for blood in order to determine the plasma, glucose, and cortisol for the physiological condition, and plasma peroxidase, complement, lysozyme activity, and total Ig content for the health and immune status. Now let's move to the results. So the results will be presented on 3 big parts: the survival and health status, the physiological condition, and the immune system status. And in each part, I will present both salmon, smolt, and eel results. So you have the kind of fish right here, so you can follow which one of them. And I will go on like that for all the results. So for salmon smolt, the recovery rate of fish was 71% and the survival was 97%, with external damage of 7% in the HPP group. In the control group, recovery was of 94% and the survival 100%, and no external damage at all. So we have better survival rate in control group and no external damage, but in HPP group, we have mainly minor injuries that are scale losses with or without hematoma. In European eels, we have the results here for gromalad, and down you have the result for ondensites. So as you can see, we had more loss, so a lesser recovery rate at ondensites and a higher mortality rate in ondensite compared to gromalad site and to the control groups. We have in both sites a better survival rates in control group and no external damage at all in control group, but we have external injuries in HPP groups that are mainly bruising, hematoma, and laceration. And the external damage rate is 8% for ondensite and 25% for HPP gromalad site. For the X-ray that were performed 6 days after, we had internal damage in both group, control group and HPP group in gromalad sites, but no internal damage at all in control group in ondensite, and 43% of eel in HPP group were showed internal damage, mainly vertebrate compaction, fracture, and sclerosis. For the physiological condition, so I come back to the Atlantic salmon. I present you the result of cortisol and glucose, and as you can see, cortisol remained stable over the time without any effect of the sites of the turbine. But glucose level decreased depending on the treatment and the sampling time. And the decrease was sharper in HPP group that are in red compared to the control group that decreased only between 72 and 120 hours. In the same registry, we prospected the gene expression for HPC70, but it didn't vary at all depending on time or the treatment. And the other genes that are related to metabolism here, carbohydrate metabolism or lipid metabolism, didn't show effect depending on the treatment. So no effect at all of the passage of turbine, but the carbohydrate metabolism genes showed an effect over the time with higher value, mainly an increase for 120 hours post-injection. And for the gene, the hormone regulation related genes, we also have an effect of time with a general increase, as you can see, at 120 hours post-injection. So for eels, for cortisol and glucose, we see that cortisol levels decreased at 145 hours post-injection in both sites. And glucose level varied depending on time and on the treatment in both sites, with a decrease of the levels during over the time, but the level remained higher in HPP group in both sites. So we can move to human system status. And here we can see that peroxidase levels were similar in the both first times in Atlantic salmon, but at 120 hours post-injection, they were higher in HPP group than control group. And for ACH complement activity levels, we can see that at 24 hours post-injection, the levels were higher in control group. They decreased over the time. And for HPP group, they remained stable between 24 and 72 hours, but they decreased between 72 and 120 hours post-injection. So the decrease was delayed compared to the control group. And the human system related gene expression showed an increase for IgM over the time only in HPP group, the red ones. No effect at all for complement activity gene expression, and an effect depending on the sampling time with a general increase at 120 hours post-injection. For lysozyme G, the lysozyme gene expression increased between 24 hours and 72 hours post-injection only in HPP group. And MPO, that is related to oxidative stress defense, was higher in HPP group compared to control group at 72 hours post-injection. For the eels, the complement activity decreased in gromalate site at 144 hours post-injection in both groups. And the lysozyme activity decreased only in HPP group in gromalate site, but it decreased in control group in onden site, with a higher lysozyme activity in control group at onden site at 24 hours post-injection. For peroxidase activity, we have a decrease throughout the time in the control group, and we have a higher level at 24 hours post-injection in control group in onden site only. We have no effect at all in gromalate site. And the total Ig content was lower at 24 hours post-injection in gromalate site, in control group in gromalate site, and increased throughout the time only in control group. It remained stable for HPP group, and at 144 hours post-injection, this total Ig content was higher than HPP group. So now let's move to the discussion. So now I will present you the result of the discussion about the survival rate and health status. So actually, the survival rate in our experimentation for Atlantic salmon was of 97%, and it was consistent with survival rates that were observed in Kaplan turbine that varied from 64 to more than 95%. And we observed mainly in the injuries large scale losses, and those large scale losses may reduce the osmoregulation ability of the fish, especially when it reaches the ocean. And it can be one of the causes of the delayed mortality. For eels, the survival rate was above, for both sites, above 80%, which is consistent with survival rates observed for Kaplan turbines. But in gromalate site, the survival rate was higher compared to onden sites, which can be the effect of the lower head turbine and the lower velocity in grommet sites. However, we observed more external damage in grommet sites, 25% versus 8% in on-den site. It can be linked to the severity of injuries. In fact, we had more severe injuries in on-den site, leading to mortality with fish that were alive after the passage through the turbine, but it led to mortality in the 2 hours of observation before putting fish back to the tanks. So the damage were more severe in on-den sites, leading to a higher mortality rates, but in the same times, it decreased the external damage rate. And we observed in on-den site more internal damage in HPP group compared to control ones, which can be related to the velocity of the turbine that was higher, and also the higher heads. In the same time, in grommet site, we observed internal damage in both control and HPP group, and it can be linked to the quality of the fish used during the experiment. For the physiological condition in Atlantic salmon smolt, the cortisol level remained stable. We know that during the smoltification, Atlantic salmon smolt have an enhanced sensitivity to stress, so they can reach the saturation in cortisol level very quickly. So it can be linked, the fact that there is no effect at all on cortisol level of the passage through the turbine on cortisol level can be due to the fact that the global stress due to the injection itself, because we need to fish the fish, to put them in a wetted tube and recover them by a net, and after we need to separate the dead one from the injured one from the alive non-injured ones. All this is a very stressful situation for the fish, and all this stress may overshadow the stress due to the turbine itself. We have more abrupt decrease of glucose level in HPP group. It can be linked to the fact that when fish face the turbine, they need more energy, so they increase their energy expenditure, and it can lead to a possible exhaustion. We observe a global increase over the time for metabolism and hormone regulation related genes. The majority of those genes are related to compensatory mechanism to glucose consumption. And as we had a little bit of stress and for everybody, and in the same time, the fish were not fed after the experimentation. So all those compensatory mechanisms to maintain the glycemia were triggered in all groups. And for the hormone regulation genes IGF-1, IGF-2, and GRH1, they were not affected by HPP. And those genes are linked to the smoltification, the beginning of the smoltification process. It can be a good news because it may mean that the ability to undergo the smoltification process seems to not be so disrupted by the passage through the turbines. For the innate immune system status in Atlantic salmon, the peroxidase activity and MPO gene expression were higher in HPP group, and those are linked to oxidative stress response. So we had probably a protection of oxidant radicals, and so we had occurrence of oxidative stress, and those peroxidase was produced in order to face this stress. The complement activity was lower in control group at 24-hour post-injection, and it decreased earlier than in HPP group. And in the same time, the lysozyme gene expression was higher at 72-hour post-injection. So we had kind of disruption of the innate human system with maybe a transient immunostimulation occurring in the beginning of due to the stress and observable in 24-hour and 72-hour post-injection. So for eels, glucose level are higher in HPP groups and decreased over the time. So it is the same conclusion than in the salmon. The passage through the turbine seems to affect the energy expenditure and trigger mechanism that are preventing hypoglycemia. And the glucose level that we observed were twofold higher in on-den site than gromalate site at 24-hour post-injection. It can be due to the fact that going through a higher water head turbine was a more stressful situation for the fish, leading to more consumption of glucose. For both groups, HPP and control groups, and both sites, we have a decrease of cortisol over the time. And we know that cortisol clearance can occur very quickly, even if the fish is experiencing stress. This clearance of the cortisol after the stress triggered probably the decrease of glucose levels. And we are addressing the issue that as the whole experiment is very, very, very stressful for fishes, maybe the cortisol levels are a little bit less relevant to assess the stress because they go up very quickly and the clearance occur very quickly after. Complement and lysozyme activity decreased over the time in both groups, and it can be due to a transient immunostimulation due to stress that is followed by maybe a recovery at 144-hour post-injection. The lysozyme activity was lower at 24-hour post-injection at on-den sites, so it can be linked to the high head turbine stress. And the passage through the turbine affected the peroxidase activity and total Ig content. So this stressful situation seems to be leading to an inhibition of some of the mechanism of the human system and oxidative stress defense. We know that stress can affect a human system by inhibiting it or immunostimulating it. So what we have to take home as a message, it is that we have some clue actually that can maybe explain the occurrence of mortality after the passage through the turbines 3 days or more after the passage itself. So this delayed mortality that was observed in the telemetric study, but not into the direct injection ones, can be explained by some disruption of the stress condition of the metabolism of the innate immune system that are linked to the passage through the turbine. This disruption seems to lead to a possible exhaustion of the fish and even a greater vulnerability to pathogen because their innate immune system is disrupted. The impact is greater for bigger species, and it is already known in terms of survival rate and in injuries, but bigger species have more mortality rates and more injuries after the passage through the turbine, and the impacts seem also greater in the midterm impacts. And the turbine types, as what already known in the literature, aggravate really those impacts in eels. So we have all those impacts right after the passage through one turbines. And as I showed you previously, in the Wallonian part of the Meuse, we have already 6 turbines. So what we address as issue is what about the cumulative impact of many passages? Because fish may survive the first passage, but what about the second, the third, and the last ones before going escaping into the ocean? Thank you all for your attention.
Thank you very much, Iman. You have still a bit of time to pose your questions in the chat function. We have already one question which comes from Dominique Couret, who is asking, at which location and where exactly did you inject the fish into the turbine? So was it near the hub, near the periphery, or mid-blade? And did this vary between the 2 sites?
So in yeah, you can hear me. I was wondering if I shut down my mic or not. So in Grand Malade site, the fish were injected really near the periphery to have the worst scenario. And in Anden site, the location wasn't very accurate because we didn't get the opportunity to send divers to put the tube in a very known, really known location. So I cannot really tell you where it was injected for Anden site, but for Grand Malade, it was injected near the periphery.
Okay. Are there any more questions to Iman?
I think no.
I don't think so.
It doesn't seem to be the case. In any case, we can have more questions after the presentation of Damien.
Yeah. So we come then to the talk of Damien Sony, who is a fishery biologist who founded the company ProFish, who is specialized in the monitoring of fish ecology. And he followed European silver eels with acoustic telemetry along the Meuse River.
Thank you very much. Hello, everybody. I will just share my screen. I hope it's okay now. I hope you are all comfortably installed in your office or maybe in your sofa or your bed, because I invite you to jump now in the cold waters of the River Meuse to track silver eels migrating along 6 successive dams that are equipped with hydropower stations. So we'll study here the global kinetics of the migration, the fish passage repartition on each site, and the first results of the mitigation measures that have been taken within the Life of Fish project that is behind this presentation. It's also a nice opportunity for me to thank very much all the collaborators of ProFish who have done a great job on the field to reach this level of data today. Thanks to all of them, you see their names. So a bit of a context first. As you see the Belgian map, the River Meuse is coming from France, crossing Belgium, and then flowing into the North Sea via the Netherlands. And usually in Belgium, we used to separate the River Meuse into 2 stretches. The upper one coming from the French border to Namur, and the lower River Meuse from Namur to the Dutch border. And this is the one that is considered here. So you see the rectangle, this is the lower River Meuse, more than 80 kilometer long stretch. The river has been totally concrete channelized for navigation purposes, and the word is not strong enough, I would have to say destroyed by the concrete more than channelized. And 6 power stations are equipping the 6 dams on this path. And consequently, of course, from the more than 10 migratory species that we used to have in the river more than 1 century ago, only 2 migratory species are left in Wallonia: the Atlantic salmon smolt that has been reintroduced about 20 years ago now, and the European eel. So since about 15 years now, we see in Wallonia arriving fish protection requirements in the renewing processes of permits under the pressure of the European Water Framework Directive and also the regulation of eels. So this is happening, in fact, side by side at the moment where a power station has to renew its permits, and that's the moment where the authority can give some new protection requirements for hydropower. So the approach is now consequently side by side, but the ecological context of each site is of course different, and normally also due to the tributaries contribution. For instance, we know that more than 90% of the population of salmon smolt will pass only through 2 turbines of this reach of the river, coming from the Ourt river, who flows to the Meuse nearby Liège. For the eel population, we don't know that much, in fact, about the real distribution and the remaining population of eels into the Meuse basin in Wallonia. This is very poorly studied and poorly documented in our region. What we know only is that the recruitment at the border with the Dutch Meuse is almost zero. It's only a few hundred individuals arriving each year. So probably there is no more recruitment of wild yellow eels, young eels, sorry. But we don't know how is the residual adult population into the River Meuse catchment. So now the context of the Life of Fish project has been introduced in 2017 by Luminous and its 4 partners. So as mentioned by Robert Mandiki, Universities of Liège, Namur, the R&D team of EDF, and Profish. The aim, saying shortly and simply, is first to meet the permit requirement for Luminous, so to fill with the protection of the fish that is demanded by the permit, but to pass from a side-by-side approach to a global river catchment approach. So to consider the whole 6 dams together with the input and output of each population, salmon and eels, and to reach a survival of about 80% for silver eels along this stretch of the river, and a survival of 90% for salmon smolt, again, considering input and output into each section of this stretch of the river. The second aim is to develop fish protection methods with a target maximal incidence of 5% loss on the total power production. So Luminous accepts to lose a little bit of production in the benefits of the fish protection. And then the third aim is to ensure that the solution that we will develop will be transferable, will be useful for others. So we have to think about that and also to communicate, which is also the aim of this webinar today. So when the project has been introduced in 2017, we already anticipated several axes of solutions. The first axis was the development of prediction models for migration of eels and salmon to reduce or shut down totally the turbine at that moment. The second axis was to develop fish passage facilities like bypass, like adapted spillage. And the third axis is to use barriers, behavioral barriers, to optimize fish passage towards the fish passage facilities. That was the axis that has been identified at the beginning of the project. Yes, at its beginning. And so the processes of the Life of Fish project is as follows. At first, just before the start of the Life of Fish project, Luminous conducted a reference stage study that has been performed in 2017-2018. And this study had 2 main axes. The first one was to assess the mortality of eel and salmon passing through turbines. So this has been done by the technique that Iman just showed you. So injection of fish into the turbine and we capture downstream with a net. So I will not speak about that today myself. The second axis of this reference state study was to also assess the eel and salmon passage repartition at each dam observed by acoustic telemetry in order to see what was the behavior of the fish in absence of any protection measure set in place. Then this was the reference states. Then the second step of the process is to select and implement the pilot solutions and study their performance on the fish passage repartition in comparison with the reference states. This is the object of my speech today. And then a future step will be to select the solutions, the best solutions that have been tested, to then deploy them at the global scale at the 6 sites, selecting which technology will go at each site in order to cover a global efficiency to reach the targets at the global scale. So today I'll present the results that we have obtained during the chapter 1B, so fish passage repartition during the reference state, and also comparison of this when the pilot solutions have been implemented. A bit of materials as methods now. So the technique that we have selected to track silver eels and salmon in the River Meuse was the JSAT acoustic telemetry. This technique has been selected after several performance tests of different technologies, and the JSAT technology has been designed especially to be able to detect acoustic signal in noisy environment like the vicinity of turbines. So it appeared to be real. This is the only technology that was able to successively bring convincing results for us. So a total of 75 hydrophones have been deployed along the 6 HPP dams and between of them to have also control station downstream each site. Additional to that, 2 sites, the first one, Grand Manade, and the fourth one, Ramay, have been equipped, as you can see here, with supplementary hydrophones to create 2-dimension possibilities to track eel behavior nearby these 2 sites, because it was anticipated already at the reference study that these 2 sites would become pilot sites to study the effect of behavioral barriers. The eels that we have tracked have been mainly obtained by a professional fisherman from the Rhine River, and I profit of the opportunity to thank again the German authorities who allowed us to bury silver eels in an official procedure to a German fisherman. But last year in 2019, we also obtained a significant amount of silver eels from our own silver eel fishery developed in the River Meuse, which was never developed before. So a total of 290 silver eels have been tagged in total of the 2 years with the acoustic tags, and eels have been released in the different stretch of the river, trying to empower each site with a significant sample to be able to reach study objectives. So go to the results now about the reference states. You see here the global kinetics of eels. So I don't know if you can read it correctly, maybe it's small, but the x-axis is the time. So it goes from October 17 to March 18. And you have on the y-axis, you have the river continuum. On the top, you have the kilometer 0. And at the down you have the last dam, which is Lix at the kilometer 81. So you see that all of the eels have been released beginning of October, and most of them waited the river peaks. You see these 4 successive river peaks in blue to go downstream, and you see that a lot of them migrated straight downstream during the peaks of river flow. The 2 last peaks of the river didn't seem to contribute a lot of eel migration, but in fact, almost all of our eels were already gone. So the system was not poured enough with enough silver eels, tagged silver eels upstream for the last peak. And so we don't know if this is an effect of a real effect that eels do not use this last peak of migration because already 3 of them was existing, or if it is because no more eels was still available in our study to use this peak to migrate downstream. About passage repartition, you see for each site from the upstream to downstream, sorry, you see a number of eels that have been detected, number of eels passing over the spillway through turbine, and unfortunately a lot of eels could not have a clear determined passage because of some overlap of some hydrophones at some places, and some technical issues that we were not able to address during the survey. So depending on if you consider that the undetermined passage are turbine or dam ones, you can worsen or not the escapement range. So we decide here to present you the escapement range from the worst case to the best case. And so you see that the escapement range at each site is quite more than 50% at all sites, and at some sites typically leaks. The last site was you have an escapement which is very, very high. Naturally, eels migrating in the high peak of river discharge, of course, naturally most of them are passing over the spillway. About behavior, eel passage are very fast. Our detection range of the hydrophones was, in fact, you can imagine it as a band of about 30 meters wide across the river, just upstream the dam. So the detection range is not that high. And so between the first detection and the last detection, of course, the fish has to migrate over something like 30 meters. So that's normal. That's finally the time between the first and the last detection is quite small. The median time lapse for all detection was 1 minute 70 seconds. But you see that the turbine passage median time was 18 minutes, while the dam passage median time was 1 minute. So eels are going straight over the spillway, but exhibit some reluctance maybe to pass through turbine, but finally are going through quite quickly, and we don't see many behaviors, many observation behaviors, except during the first days after the release. As you mentioned, eels were coming from the Rhine River, has been tagged and translocated in the River Meuse. So during the first days, you have sometimes some explorating mobility of eels that are arriving nearby the dams, and this is not clear if it's a migration attempt or an exploration behavior, but this was in very low discharge anyway. So about the dial distribution of the passage, again, it's probably quite small, but you have all the 6 sites and it's the time of the day. So you start from 0 hour to 23 hour, and you see that you have from upstream to downstream, you have a strong nocturnal pattern on the first site, and then a shift towards the 3 downstream sites. You see that more and more eels are passing during both night and day. And if you see the graph here on the left, it's the graph that is pulling together all the first sites, all the eels all the first passage, if you are saying, or in other words. So then you have a very strong nocturnal pattern. So it seems that once eels start to migrate, when they meet the first site, it's a very strong nocturnal pattern. And then they go straight down, and we see that eels are making the whole migration of 80 kilometers on time. The shortest, the fastest one was something like 18 hours to do everything. Of course, if the eels is starting in the evening, 18 hours bring the eel midday the next day. So they start at the beginning of the night, and then they go downstream, straight downstream until maybe through the day after, or maybe a few days after, because of course our survey stops at the Dutch border. And so it's important to consider if the origin of the fish can maybe affect the time of passage And of course, the study here might influence the result. Okay, so now that was for the reference states, and now let's just quickly see what kind of mitigation measures have been selected by Luminous. So Luminous sorted the market, make a request, European request for proposal for different possibilities, and selected himself the behavioral barrier. So an electrical barrier was installed in the first power station in Grand Malade, also called Namur. Basically, it consists in a stainless steel pipe anchored to the bottom and floating in two lines, one positive, one negative line. That was the system installed by Procom System last year in Grand Malade. On the second site, Anden Power Station, we decided to test the application of yield prediction model developed by the team of EDF R&D into the Life for Fish project. So globally, the patterns, the variables used by the model are a global time frame between 20 August and 28 of February, a river meuse above 200 cubics, and then when these two conditions are met, we look for the gradient of discharge on the last 5 days, and when gradient is over 35 cubics, it induces a turbine shutdown for duration of 12 hours, 6 p.m., 6 a.m. Already 2 publications have been made for this model, so you can find here the reference. And the third system was developed and deployed in Ramey. It's a bubble barrier developed by a company called Apumas that has been installed 10 meters away of the intake of the Ramey power station, beginning of October. So after the installation of the system, then we perform a new telemetry study to compare the behavioral passage of eels with the reference states that I just presented to just before. So in the second survey, we also had a quite good detection range, 80% of detection range. And again, as you can see, eels are migrating with the same kind of patterns than 2 years ago, using mainly the peaks of the river to go downstream. So it was quite similar, and globally, the hydrological conditions compared for both years was not significantly different. So the global conditions were the same for both years, which is a good point when you want to compare behaviors. So focusing now on the first mitigation measures, which is the electrical barrier in Namur. Here you have the comparison of the escapement rate in Namur for 2019, when the barrier was there, with 2017. As I said, so you see that escapement is improving, but as I said, globally, the discharge condition over 2 years was not different. But if you look for the discharge condition associated for each eel passage, the conditions were a bit higher in 2019 than in 2017. So this is also a fact that finally in 2019, both the presence of the barrier, but also a bit higher hydrological conditions can influence a better escapement, excuse me. So to try to avoid the effect of the hydrological measures, conditions, we developed, as it has been made by a lot of French colleagues before, we developed a model of logistical regression. So saying it simply, in the y-axis, we put all the turbine passage as a 0 value and the dam passage as a 1 value. And on the x-axis, you have the ratio of discharge over the spillway compared to the discharge of the river. So at 0.5, you have 50% of the river going over the spillway, 50% going into the turbine. You see that at 0.5, in red, you have the escapement rate model of the reference state. And you see that when the river is split 50-50 between turbines and the dam, you have an escapement rate reaching 70% naturally. When you see that when most of the river is going through turbine, the escapement rate is decreasing a lot. And the same situation is observed for 2019 in the presence of the barrier, and you see that it's funny to see that the two lines are crossing exactly each other at the ratio 0.5. So meaning that above this ratio, the escapement was not different, but below this ratio, in other words, when the discharge is mainly going to the turbine, we had better escapement rates observed in 2019 than in 2070. And you see the difference is unfortunately not significant, but you see the p-value of the Mann-Whitney test is, let's say, slowly close to significant. So we, in the report that will be soon available, we tested different analysis. I will not present all of them here, but globally, the presence of the barrier clearly induced an improvement of the yield escapement that is also independent of the discharge. The maximal quantification estimates a reduction of turbine passage by 52%. But as I said, there is a slight influence of the discharge condition. So probably the real range efficiency of the barrier is between 40 and 50%. So this is not a wonderful novel, but it is enough for us, for the Life of Fish project, to consider the system to be kept in the toolbox for the future, because as you have seen, the natural escapement rate of the eels over the spillway is already high, and with an efficiency of 40 to 50%, it's enough to reach the target protection we need to reach. And finally, the barrier also resisted quite well to the winter discharge and debris, so the crash test also was revealing to be satisfying. That was for the electrical barrier, and then go down to the next site in Ardennes, where the turbine shutdown model was applied in last year. So we removed from the dataset all the eels that were detected less than 7 days between the release and the passage in Ardennes, because it is the same condition that has been, let's say, posed by the model when the model was attributed to try to remove any behavioral effect due to the tagging procedure. So removing this, we only have 44 eels passage left during the time frame of the operation of the model, and during the global time frame of the model, 32 shutdowns alarm have been generated, inducing normally turbine shutdown. From the eels that we have passed to Ardennes during that period, 21% of the eels passed out of the date of prediction, so totally out of the dates. 80%, close to 80% of them passed during the right date, and within the right date, 50% succeeded to pass. I would say the model succeeded to predict 50% of the eels passing into the right time frame, 6 p.m., 6 a.m. But in practice, the shutdown had not been performed successfully. So when the model triggers an alarm, you have a procedure inside the control room of the power station, then a communication procedure with the Walloon authorities of navigation, because it's not the same authority that drive the dam and the power station. And so for many, and it was the first time that this kind of procedure was also installed. So most of the time, the turbine shutdown was not performed successfully. So in fact, 70 eels, about on the 22 that succeeded to pass in the 6 p.m., 6 a.m. time, 17 of them passed during ineffective shutdown. So, but all of them passed over the spillways, meaning that if a shutdown would have been made, none of them would have been saved because it was already saved. And only 5 eels were passing during effective shutdown. So that was finally a very small sample study. Excuse me, I'm going too fast. So in conclusion, for the turbine shutdown, in its present version and applied in Ardennes, the prediction model reached 50% in the 6 p.m., 6 a.m. time frame. So effective shutdown in the situation of Belgium is tricky because the turbine operators do not handle the dam operations. in other countries or on the other regions. I know from experience that both dams and turbines are operated by the same company, so it might be easier for them to implement that. An effective operation of the model in Andenne could have developed maximum 1 eel from a turbine passage to a spillway passage. So in fact, Andenne is a site where we have naturally a very high escapement rate over the spillway. So maybe it was not the best site to study the effect of this system. Remember that there is a possible alteration of the eel passage time we observe due to the weight of the sample repartition upstream. As I mentioned, eel are starting beginning of the night, but here is the second site, and so maybe what we observe is not exactly the reality of a wild migration. And then so I think that the model should be tuned, adjusted with data observed on wild migrating eels with passive acoustic observation, for instance. And then the third mitigation measures was the bubble barrier that has been commissioned on the 20th September, but unfortunately did not resist the winter discharge, and so the system was destroyed by the end of November. So on that short time frame, only 15 eels were passing close to the barrier, and all of them went through the turbine. We have 14 2D tracks of eels available, and we didn't see a major or obvious eel reaction close to the bubble barrier. So in conclusion, the barrier is not considered as a future solution in the toolbox because mainly of a lack of reliability. We didn't have enough data to validate it on the behavioral point of view, but the first data are not so encouraging neither on the biological effect. So arriving to the end of my speech progressively, conclusion, among the solutions tested, the electrical barrier and the eel prediction model are the two that are considered as available in our toolbox. Next step for us is to decide what system to implement and where, and do we implement them alone or together in association on different sites. So we have now to consider each site, the ecological global weight of each site, and see where we will put the highest protection level and where we have to put the lowest protection level. So these tools are increasing the escapement rate over the spillway. So the effect of these two tools is to send more eels toward the spillways. And so the next autumn, 2021, will be the period where we will start the third and the last silver wheel tracking with telemetry to check the global efficiency of the measures deployed on the entire scale, so the 6 sites. So this will be for next year. So soon you will probably have the data of the final solution for it. Just a few words for smolt. Next springtime, we'll test also the pilot solution for smolt. It's been delayed by 1 year due to the COVID crisis. So we'll test the efficiency of a bypass and the efficiency of the electrical barrier as a guiding tool to the bypass. We'll test also the efficiency of a smolt migration prediction model, and also an adapted spillage at the site of Ivorame. And just before to thank you for your attention, I just raise a crucial question that we observed. As I mentioned, the aim of our solutions is to send eels and salmon, mainly eels, over the spillways. But are the spillways a safe route for silver eels? During the last survey, 47 eels have been reported missing. So we're waiting for them at the site, but they did not arrive, probably because they stopped or they died during their migration. And so we look for the last passage before to disappear. And you see that 51% of the eel passage was over the spillways, while 38% over turbines. So what we have done with our data is to pool together all the passage, turbine passage and spillway passage. And so looking for only all the turbine passage, we have a disappearance rate of about 22%. And so we should say, okay, 22%, this is consistent with the expected mortality range induced by the turbine. So we should consider as normal mortality induced by the turbine. Then we have a total of about 10% of disappearance rates due to spillway passage. So we might be tempted to say, maybe it's predation, maybe it's something else, but I think it is also probably an effect of the dam. There is something happening when an eel is passing over the spillway. I'm not the first one to tell that. The literature also starts to bring some information like that. So I think that a safe dam passage might be the next challenge for migratory fish in the next decades. So I know it's late. I hope you are not so sleepy now. I thank you very much for your attention.
Thank you very much, Suni. We have already one question. What about the 2D individual trajectories in the presence of the electrical barrier? Did you see rejection behaviors that would support a barrier efficiency?
Not clearly. Yes, you might be surprised because I mentioned the 2D tracks and 2D network to study eel behavior, and finally I do not show any track in Romalade. Well, in fact, eels were passing very fast, as I mentioned, and it was a bit, most of the time, too fast to have a clear tracks of eel migration. We have mostly a lot of geo-reference spots, but not clear tracks of eels. So we have no obvious reaction, neither of eel behavior close to the electrical barrier, but we also have no obvious, no very clear tracks of eels going to the spillway or going to the turbine. So this is not, the 2D tracking there is tricky with silver eels because they are swimming too fast in high fluid condition, which is the condition where the hydrophones are the most impaired in their acoustic capacity. So the detection range is also reduced during high fluid conditions. So not very obvious, yes.
Okay. I have a question concerning the barriers that are introduced. So you said you will apply the electrical barrier again and the prediction model. What about the economic factors? So what are the costs of these barriers that are introduced?
I'm not very, I do not have this precise information because the purchase procedure was something depending on Luminous. So I invite you maybe to send me this question by email and I will forward that to Luminous because I'm not in position to tell that to you. I didn't participate to the selection of the candidates. So I don't know exactly. But what I can tell you is that we have to consider now the cost effectiveness of each measure. Of course, the good point with the barrier is that you succeed to deflect eels without stopping the turbine. And the comparison with that is to stop the turbine for maybe up to 20, 30 nights without installing nothing, without any investment. And so these two systems needs to be compared on the cost effectiveness and also in a long-term view, because you might have some maintenance for the futures for the barrier, for instance. You have also good years and bad years in hydrology and for turbine production. So this is a step that needs to be addressed now, is the cost efficiency on the economic point of view too, yes.
Okay.
Caroline?
Yes.
Yes, I think that there was also a question from Alain Gillet about statistics on the presentation of IMEN.
Yeah.
Yeah, so to come back to the question of Alain Gillet about the fact if the difference between the different groups are statistically tested, yes. The little letters that were displayed on the graphs indicate significant difference between groups. And I also had a question from Mr. Nzo Matandobili about the injection hour, if it affects or not the response of eel for the observed parameters. And actually we did exactly the same procedure of injection. So we began at the same time and we didn't have so much difference in the injection hour between both sites. neither in the injection period, because we did the experiment nearly in the same period year after year. And for all my data, the paper concerning the salmon is already published, and the paper concerning the eels is already accepted, so the publication will be soon public, so it will be okay to have more information on the basis of the paper. We have two more questions for Damien. Caroline, I leave you to...
Okay, so one question is, could you give some information about barriers resistance to debris or high flow conditions? Is there some damage occurring during floods, for example?
Well, I was expecting much more trouble to the electrical barrier than we observed. In fact, yes, the electrical barrier resisted quite well. So you see that these vertical electrodes that are bottom anchored and supported by buoys, so they have the capacity to lay down. And then, for instance, when you see the turbine starting, the currents and the water velocity put them already a bit in a slope due to the effect of the current. But no big damage arrived to them. I guess that a lot of debris are now lying on the concrete, on the anchors of the systems. But I would say mechanically the system is still in operation after one winter. But the situation in this sites, Grand Malade and other places in the Luminous power station, is that the power station are like an additional gate to the dam, so it's a bit lateral to the flow. And when high winter flow condition, in high flood condition, the head of the dam becomes too small and the turbine will shut down. So in very high condition, in fact, the area where the barrier was very quiet, very still, because there is no more discharge to the turbine, and you have a storm discharge into the main river that will maybe spare the exposition of the electrode to very big debris. But that happens when it is more than 600-700 cubic, so you have a lot of debris before that, and it resisted quite well. The bubble barrier was not resisting that much, as you have seen. So I have no real explanation about what was broken and why. But the fact is that the system was quite quickly broken.
Okay, and there's another question for you, Damien. Whether the bubble barrier were tested, the bubble barrier were tested under which, what kind of flow conditions? A wide spillway discharge? I don't know what that is.
With spillway discharge, yes. The 15 eels that have been observed approaching the bubble barrier were approaching in condition where you had a majority flow over to the turbine and a small flow over the spillway. Typically, so the spillway was not very attractive for eels at that moment. But in same situation of low water on the spillways in Grand Malade, we have an escapement of eels that we didn't observe in the reference state. So yes, it's not very conditions that are perfect for eels passing over the spillway, but it was open a little bit.
Okay. So thank you very much to the speakers. I don't see any more questions. So yes, thank you very much for your talks, and thanks all the participants or the public who listened to you. And well, have a nice afternoon and see you next time. Bye bye.
Bye bye.
Bye bye.
Bye.